Inflammation in Parkinson's disease, Mechanisms and Therapeutic Implications. Abstract. Parkinson's disease (PD) is a common neurodegenerative disorder primarily characterized by the death of dopaminergic neurons that project from the substantia nigra pars compacta. Although the molecular bases for PD development are still little defined, extensive evidence from human samples and animal models support the involvement of inflammation in onset or progression. However, the exact trigger for this response remains unclear. Here, we provide a systematic review of the cellular mediators, i.e., microglia, astroglia and endothelial cells. We also discuss the genetic and transcriptional control of inflammation in PD and the immunomodulatory role of dopamine and reactive oxygen species. Finally, we summarize the preclinical and clinical approaches targeting neuroinflammation in PD. 1. Introduction. Parkinson's disease (PD) is a common neurodegenerative disorder primarily characterized by the deterioration of motor activities due to the impairment of the dopaminergic nigrostriatal system. Specifically, the death of dopaminergic neurons that project from the substantia nigra pars compacta to the caudate putamen in the striatum results in the loss of dopamine neurotransmission, causing the primary motor symptoms, including tremor at rest, bradykinesia, rigidity and postural instability. Although PD was initially described as a movement disorder without dementia, it is now accepted that PD progression affects other extranegral dopaminergic, cholinergic and serotoninergic tracts, leading to non-motor symptoms that include anosmia, sleep disorders and constipation as well as cognitive and psychiatric symptoms, such as dementia and depression. The onset of the disease occurs decades before the first symptoms appear. However, the ultimate underlying causes of dopaminergic death remain unknown. While 5 to 10% of PD cases are of genetic origin, mutations in the PARK genes encoding alpha-synuclein, DJ1, PINK, LRRK2, etc., leading to early PD onset, most cases remain idiopathic and are associated with aging. In addition to genetic predisposition, there are risk factors associated with environmental toxins, pesticides, heavy metals, traumatic lesions and bacterial or viral infections, all of which are closely associated with inflammation. A crucial role of inflammation in developing Parkinsonian symptoms has been suspected for many years since the initial observation of Parkinson-like symptoms in individuals infected with the influenza virus Encephalitis lethargica. Thereafter, PD onset has been associated with several viral pathogens including influenza A, herpes simplex virus 1, HSV1, Ebola virus EBV, varicella zoster virus, Japanese encephalitis virus, vanilla necrosis virus, human immunodeficiency virus as well as with Helicobacter pylori. Neurotropic pathogens might reach the basal ganglia through the nasal mucosa, via the olfactory pathways, and through the intestinal mucosa, via enteric plexuses and preganglionic vagal fibers, ultimately leading to a cascade of neuroinflammatory and neurodegenerative events in the nigrostriatal tract. It is interesting that, in addition to eliciting an intense immune response in the nigrostriatal tract, some viral proteins HSV1 and EBV exhibit molecular mimicry with alpha-synuclein alpha-syn and promote alpha-syn aggregation and further accumulation in intracellular deposits called Lewy bodies. Moreover, in the enteric nervous system alpha-syn exhibits potent chemoattractant activity of neutrophils and monocytes in response to viral infection, further implying this protein in systemic and brain inflammatory responses in the pathogenesis of PD. Inflammation is a highly regulated mechanism against pathogenic stimuli or tissue injury, aimed at protecting the host from the damage-causing agents and to promote tissue repair. The central nervous system, CNS, has long been considered an immune-privileged tissue due to the separation from the peripheral immune system by the blood-brain barrier (BBB). However, this concept has been revised. In fact, a potent innate immune response against pathogen-associated molecular patterns (PAMPs) and damage-associated molecular patterns (DAMPs) can be elicited in the CNS. Under physiological conditions, microglia and astroglia constantly survey the brain parenchyma to maintain CNS homeostasis by releasing neurotrophic factors, removing synaptic glutamate, remodeling and reshaping synapses, etc. However, these glial cells can be activated by PAMPs and DAMPs, such as secreted factors from damaged neurons or protein aggregates, leading to persistent neuroinflammation. Although it may not represent an initiating factor in all case PD, chronic neuroinflammation appears to be a cofactor for disease progression. As further discussed, extensive evidence in human samples and from animal models support the involvement of inflammation in the development of PD. However, The exact trigger for this response remains unclear. While inflammation might be the consequence of ongoing neuronal cell death in PD, it is also likely that misfolded alpha-syn might play a direct role. In addition to the well-documented microgliosis and astrogliosis in PD brains, peripheral inflammation and PD risk-associated genes support an important contribution of the chronic inflammatory response on the progression of this neurodegenerative disease. We will provide a systematic review of the cellular mediators and molecular mechanisms involved in neuroinflammation and how they may impact PD progression. Concluding remarks: chronic inflammation can directly or indirectly contribute to the etiology and progression of PD. A possible scenario is that chronic inflammation in the olfactory tract and/or the enteric system due to environmental and pathogen exposure throughout life and/or alterations in gut microbiota favors alpha-syn aggregation and spreading to the brain, further aggravating inflammatory responses that contribute to neurodegeneration. Therapeutic intervention for PD remains an urgent challenge. Due to the central role of inflammation in PD, immunomodulatory therapies become prime targets for research. Although inhibition of neuroinflammation itself, either independently or as a combined strategy to other interventions, may not alter the underlying cause of the disease, it may reduce the production of factors that contribute to neurotoxicity, thereby resulting in clinical benefit.